Welcome, friends. Welcome to my topic, that is revision class on plant pathology, which will be useful for JRF and SRF and other competitive examinations. In this class, we are going to see about the fungi, bacteria, virus, phytoplasma, and viroids. We know that fungi constitute a major contribution to plant diseases by affecting the host plants. And these fungi are nothing but the eukaryotic and achlorophyllous, that is the divide of chlorophyll, and these are unicellular or multicellular organisms which will be microscopic or rarely macroscopic. Example, macroscopic fungi means mushrooms. And so these fungi will reproduce by asexually or sexually, by producing asexual spore or sexual spores. And in fungi, three types of structures are there, that is hypha, mycelium, and thallus. Hypha means nothing but a web-like structure, that is like a tubular structure, um, which is hair growth from the mycelium. Mycelium is nothing but the mass of hyphae, which constituting the body of the fungus. The body of the fungus, that is the entire body, is called the thallus. And while coming to the types of mycelium, there are three types of mycelium: intracellular, intracellular, and both inter and intracellular. Intracellular means if the mycelium occurs in between the cells, it is called intercellular, which will be seen in several oomycetes fungi like white rust, or that is albo, candida, or pythium, or phytophthora species. And intracellular mycelium means the mycelium occurs present inside the host cell. It is called intracellular mycelium. Example, several saprophytes like Fusarium, Alternaria, Cercospora, others. And next to coming to inter and intracellular, example, it will be seen only in Fusarium, that is Fusarium udum, or Fusarium oxysporum, or it will be seen in all species of Fusarium. Then coming to sexual fruiting bodies, sexual fruiting bodies consists of ascocarp and bestiocarp. Ascocarp means carp means fruiting body, which means ascocarp means the fruiting body which is produced by the ascomyces fungi. It consists of chasmothecium, apothecium, perithecium, and pseudothecium. Chasmothecium means that is a closed structure. Previous it is called chasmothecium, but now it is called as clistothecium because of same appearance. It is seen in all powdery mildews like Erythrophia, Ancinula, and others, Penicillium, and Aspergillus. Then coming to apothecium, which is nothing but a saucer-shaped structure or inverted cup-like structure, which will be seen in Diclochroma rosea, that is a black spot of rose. Then coming to perithecium, which is called a flask-shaped structure, which is produced by the claviceps. And pseudothecium, this is called which is produced in the Venturia inicolis, that is apple's cap, which is nothing but a flask-shaped structure, that is bottleneck structure. The difference between perithecium and pseudothecium means both are flask shaped, but the assay is arranged in a hymen layer in case of perithecium, and assay is not arranged in case of pseudothecium in the hymenium layer. These are the sexual rooting bodies produced by the ascomyces fungi, like clistothecium, that is a closed shape ascocarp. Perithecium, it is a flask shaped ascocarp. And apothecium saucer shaped, and pseudothecium also a flask shaped ascocarp. And while coming to second sexual fruiting body produced by the Basidiomycete fungi, is called Basidiocarp. So in this mushroom and bracket fungi will exist. In mushrooms are edible and non-edible will exist, like Agaricus, Pleurotus, and Amanita. And in bracket fungi, Ganoderma, which produce a bracket structure in the coconut, that is which causes tangerine wilt in the coconut. Then coming to asexual fruiting bodies, like sporangium, pycnidium, azarulus, porodokium, cinnamma, and sorus. Sporangium is like a sac-like structure where zoospores are present. We know that zoospores will be whiplash or tinsel, or maybe motile or non-motile. Pycnidium, which is a flash type structure produced by Macrohomina and Brotia diploidea, and Azarullus. We know that Azarullus is produced by Coltotrichum caps, that is all species of Coltotrichum, and this is a metaphypha giving rise to short conidiophores closely packed together. And Sporodocium, we know that it is produced by the Fusarium and a Nectaria species. It is a cushion shaped stroma. And Cinema is it's a group of conidiophores which is cemented together and forming a elongated spore bearing structure. This is mainly produced by Sartocystis species, which causing pineapple rot in pineapple pineapple rot or set rot in sugarcane. Next, sorus, which is nothing but sori in plural, which is produced by the rust and smut fungi. We can see the smut balls, or we can see a sori that is called ball. It's a ball-like structure. Then this is the asexual fruiting bodies consist of pycnidium and Azarullus and Sporodocium and Cinema. Then coming to sexual spores, sexual spore contains oospores, zygospores, ascospore, and basidiopore. Most of the students will take zygospore as a asexual spore, but this is a zygospore is a sexual spore, because just remind zygote, remember as a zygote, so which will be produced by fusion of male and female gametes. Oospores, this is a thick-walled diploid spore. In several cases, oospore is also a resting spore. Always the oospore, that is oospore will be a resting spore, which will be inoculated in the previous season of the crop, and which will be affect the upcoming season, upcoming cropping season. This is produced by Pythium and Phytophthora species. Zygospore, it is a fusion of male and female gamete or two gametangia, which is produced by Rhizoctonia. Ascospore, I already told, the spores which is produced by the Ascomyces fungi by the fusion of male and female gametes. It is mainly produced in powdery mildew, Aspergillus and Penicillium. Then Basidiospore, this is that is a fusion of male and female gametes of the Basidiomycetes fungi. Example, rust, smut, and in mushroom, Basidiospores are produced. Then coming to asexual spore, sorry, this is sexual spore, this is asexual spores. In asexual spores, there are ascospores, basidiospores, and zygospores. Sorry, sexual spores only. Later I will cover the asexual spores. In sexual spores, I told ascospore. In ascospore, which is produced the assay in the, which will be in 18 number. This is very important. The ascospores always will be in 18 number, but in some cases it will go to 4 numbers also. Then basidiospores, you can see the basidiospore in the sterigmata. Then zygospores by fusion of male and female gametangia, which is produced by the Rhizoctonia. Then coming to asexual spore, first one sporangiospores, conidiospores, arthospores, chlamydospores, and blastospores. In the sporangiospores, that is sporangiospores are produced in sporangium, that is asexual fruiting body, sporangium. There are two types of sporangiospores. One is planospores and another one is aplanospores. Planospore means it's a motile spore, which is called zoospore. Another one is called aplanospore, which is non-motile zoospore. Just remember plane, which will be moving, plane which will be flying or moving. So a zoospore will move like that. Aplanospore means the spores which is non-motile and can't able to move. This aplanospores are produced in oomocytes like Pythium and Phytophthora, and aplanospores in Rhizopus and Mucor. And conidiospore, it will be produced in Fusarium, Alternera, and Cercospora, and arthospores. Arthospore means nothing but the, if the mycelium or the hypha is there, which will be fragmented and produce the spores, like that fragmented part will act as a spore, that is called arthrospore, which is also called as arthroconidia, which is produced by the Tracheosporium and Geotrichum. Then chlamydospores, this is a resting body or the resting structure which is produced by the fusarium, which will be a chlamydospore will be a intercalary chlamydospore or intercalary intra and intercalary chlamydospore or or a terminal chlamydospore or intercalary chlamydospore. Then blastospore means nothing but the spores which is produced in the blastic action, that is in a forcible movement, in a forcible action, blastic reaction. Example, it will be produced in the mushroom. These are the asexual spore. First one we will see the arthrospores. Here in this diagram we can see the several spores are fragmenting that mycelium will be fragmented to form a spore, that is called arthrospores. Then here you can see the chlamydospore, that is terminal chlamydospore and intercalary chlamydospore. Then sporangiospore, that is a sporangiosspore which will be releasing from the vessel like or sac like structure, that is sporangium, which will be a zoospore or aplanospore. Zoospore means motile, aplanospore means non-motile. Then conidiospores, which will be produced from the conidiophore in most of the species like Alternaria, in Fusarium, that is macro and micro conidia. And blastospore, which is produced by the Basidomestis fungi in the forcible action, so it is called blastospore. Then coming to sexual reproduction in fungi. In sexual reproduction, it involves isogamy, anisogamy, oogamy, spermatization, gametangel contact, the gametangel copulation, like that. In this, most important is gametangel copulation, in which Anthridium oogonium will go for copulation for transfer of genetic material to produce offspring, that is a oospore, like that. In this, spermatization is important. Spermatization means in rust and smut fungi there is both female and male uh, gamete are there, that is a female means a receptive hypha will be there in one place, that is non-movable, that is non-motile receptive hypha, and motile male, that is spermatia. This spermatia is blown through the wind and it will be attached to the receptive hypha, and then the transfer of genetic material will happens to production of offspring. Then next method is sexual reproduction. In asexual reproduction, fission of somatic cell, that I told, that arthrospores which will be produced by the fission, by then budding, then fragmentation, and asexual spore formation. Then we have completed about some of the important fundamentals of the pathology, and now coming to history part. The first, the Romans, the uh, the Romans created a festival called Robigo or Robigalia, which is for uh, celebrated for rust fungus. And then we know that microscope first invented by Leeuwenhoek. Later, that some of the updation in the microscope will done by Jensen and his co-workers. Then we know that in microscope, electron microscope by Nolan Ruska and John Needham first observed the appearance of organisms and conclude that microorganisms originated from the meat. And other important is Micheli, who is called as he wrote a book called Nova Plantarum Genera. And Matthew Tillett, he inoculated with seeds and dust with the dust of smut. And actually smut, we will see that Tillacea carries like that, that is genus and species. The Tillacea, the genus which will that came from his name, that is Matthew Tillett. Then Prevost, he repeated the Tillett experiment and proved that burnt off wheat caused by the fungus. And he studied the germination of the smut spore and their inhibition by copper sulfate. So it is important that copper sulfate for the inhibition of smut is first done by the Prevost. Then Andenbury was called as a father of plant pathology, important. um, then oronin in some of the phytoplasma, sorry, in plasma, that is in protozoa, it just caused a disease in crucifers called Plasmodiphora brassicae, clubroot of cabbage, or yeah, mostly clubroot of cabbage, that produce a oronin bodies. So that name came from that scientist Voronin. Then TJ Burrell, he uh, he he proved that fire bite of apple and pear is caused by the bacterium Yersinia amylovara. And Adolf Meyer, it's important, he concluded that the infectious nature of sap transmission in tobacco plants for TMV. And Beijing is called as a father of virology, and he he coined the term contagium vivid fluidum for the term virus. And John Needham, Needham, for nematode, that proven that ionotriticity, that which cause wheat gall, which is called wheat gall nematode. And Doe et al., which who proved that mycoplasma or phytoplasma cause disease in potato, that is potato which is proved. And Diner viroid, which is associated with the first viroid disease, potato spindle tuber viroid. This is the smallest viroid known. And and DeBerry, he finally proved that the cause of late blight of potato is caused by the Phytophthora infestans. And J. Kuhn, he published the book Disease of Cultivated Crops and Their Cause and Control. Then Robert Koch, he posted the Koch postulate for the proof of pathogenicity. This is very important. For the proof of pathogenicity, he given the postulates, that is four postulates. Then Oscar Berfeld, he given the technique of pure culture. And we know that PMA Millardat, who is the student of the Anton DeBerry, he discovered the board of mixture for the control of downy mildew of grapes. Then E.C. Stackman, who give the phenomenon of a physiological specialization in rust. And William Brown, role of enzymes to disintegrate plant tissue. And Muller and Boger coined the term phytolaxin. And Ronald Ross, he gave the term SAR, that is systemic acquired resistance. Then coming to Robert Hattick, that is who is called as father of tree pathology, that is forest pathology. And T.J. Verrill, we already told, of, prove that bacterium causes disease in apple and pear. And Adolf Meyer. Adolf Meyer proved that infectious nature of sap transmission from tobacco infected plants. And Ivanovsky, this is sap retained infectivity even after filtration through Chamberlain filter. Chamberlain is nothing but a filter. That is, Chamberlain is a scientist who developed one filter called Chamberlain filter. So this sap, which is obtained from the tobacco mosaic virus plants, he purified using that Chamberlain filter, so that virus particles are remained in that filter. So it is first done by Ivanovsky. Then Martin William Beijering, who is called as a father of virology, I already told, he given the term contagium fluidum. And Stanley, W.M. Stanley, is very important for crystallization of virus, and he shared a Nobel Prize also. And Kasanins, he coined the term satellite virus associated with the tobacco necrosis virus is the most important. That is, satellite virus is associated with tobacco necrosis virus only, and it is also called satellite virus, also called as a helper virus. Then Clark and Adams, who coined the who developed ELISA, it's most important. And Biffin is he reported the resistance in two wheat varieties to yellow rust, which was initiated in a Mediterranean version to its progeny. Then Barres, he given the pathogen species, that is genetic variability in the pathogen. Then Floor, that we know that gene for gene hypothesis in the linseed rust. And Gauman, who gave the concept of hypersensitivity. Hypersensitivity is also called as a programmed cell death, which it means when the pathogen is attacking the plant will give some response to that pathogen. So immediately the neighboring cells in the affected portion will die, so that the pathogen can't able to infect furthermore. So then Van der Planck, he coined the term vertical and horizontal distance, and this is also called as a father of epidemiology. So he wrote a book called Plant Disease Epidemics and Control. Then Millardat, we know that he developed Botox mixture. Then Tisdale and Williams, they given diethiocarboic derivatives fungicides. We know that the first systematic fungicide is called carboxin, which is given by Von Schlimming and Kulka. Then E.J. Butler, we know that while coming to this is for Indian scientists, very important. E.J. Butler is first mycologist for India, that is father of Indian plant pathology. He written the book Fungicides and Disease in Plants. So this book is important. And Casey Mehta, monograph of annual recurrence of black stem rust of. So whenever in the exam they will ask some rust. The first rust is was first studied by Casey Mehta only. And Critiker is the first Indians to study the fungi. And Mitra, while Mehta is concerned to the rust fungi, Mitra is concerned to the bunt. Then Luthra and Satter, they given solar heat treatment for the loose mud disease management. And B.B. Munkar is the father of Indian Phytopathological Society (IPS), and he given the book. He wrote the book Fungi and Plant Diseases. And while coming to last, this is one of the historical perspective very important. They will ask when the lead bite of potato uh, will leads to overall world's that famine, starvation, that is called the Irish famine, that is in 1845 is correct. And the coffee rust from Sri Lanka, downy mildew grapes from France, Bengal famine, that is in India due to black spot of rice, then bacterial blight of rice in Bihar, then southern corn leaf blight in USA. I conclude this PPT with this, and later I will go upon the management and major diseases. Thank you.